All right, hi everybody. Welcome to Chapter 10 on nucleosomes, chromatin, and how the storage of DNA is possibly the largest gatekeeper to what genes are on, off, and at what level. So we have covered a lot of stuff when it comes to the structure of DNA, the chemical, the kind of chemical structure between what's open, closed, different conformations of DNA, things like that. Today we are going to try and go in and kind of assess each of these levels of how this works. So we're going to cover that histones are these circular small proteins. When you have 8 of them, that is a full nucleosome. DNA will wrap 1.6 times around a nucleosome. There will be an exposed region usually, and then you will have 2 more wraps here, for example, and another exposed region. You can stack these nucleosomes on top of each other, as you can see here, and these stacks are going to form large structures that you can see put right here. One big thing to notice is that physically, active DNA that is more accessible to transcription factors and then RNA polymerase is going to be more open. It is going to appear larger. When you are tightly packed, it is going to appear much less accessible. It is going to be a lot smaller. For example, this length of compact DNA is almost the same length as this open DNA, but there are 4 strands, 5 strands in this one, maybe even more in the inactive tight pack. So eventually, like when you're about to replicate, your DNA, or you're going to pull this chromosome apart at each way, you have to fully tightly pack all the DNA for this journey, essentially. So that's one of the important things. But the main thing that Chapter 10 gives us is all the ingredients for opening genes up and how to close them down. So the main thing that we talked about was what are the things that are storing DNA? And a long time ago, well, 10, 15 years, 20 years, I don't know what a long time is in molecular biology, we found that there were 5 different proteins, these histones. They are big, spherical, and they are made of tons of lysine, let me see if I can draw a K, and arginines. Both of these have massive positive charges, and they are going to basically attract the negative phosphate backbone of DNA that is going to wrap around them. And you need 8 of these. So mainly remember these ones. So we'll cover H1 in a sec. But these are the subunit types of histones that form the collective 8 histone octamer that DNA is going to wrap around. H1, we'll find out, this histone is going to occupy the region right here. That linker DNA, that's H1, that little green guy right there. Okay? So H1 is not part of the octamers. It is responsible for finding links between the DNA. And what we'll see is that if H1 is present, this is closed, as you can tell, it's kind of closed for business. When H1 can get removed right here, that DNA is then open for transcription. Okay, so one of the things that they found is that when they were able to isolate DNA in these little in these little structures, they found these 5 things that were always abundant as proteins. They also found that there was about 200 base pairs wrapped around each time. Now they also saw 400, 600, 800, and 1,000 bands. Why might that be? What they figured out is that it's just tons and tons of segments of 200 base pairs. So what they did was essentially shear the DNA into the pieces that were basically around one of these units. So the DNA that's around one of these nucleosomes, right? That usually works out to about, or at least around 200. So the 400 and the 600 and so on, that's just larger groups and collections of 200s, for example. So in this case, we found that 200 was the number of base pairs that are going to go around a single nucleosome. So that's how much is going around. Okay, so like we saw, there are 4 major subunits. H3 and H4 are proteins that are built to become a dimer. That is their quaternary structure right there. They are built to form this small subunit. Okay? This dimer is very good at forming a tetramer. So now you have 4 H3s and H4 combinations, so 2 H3s and 2 H4s. H2A and H2B, they are very good at combining into a dimer as well. And then this dimer is going to oftentimes combine with this tetramer right here in some combination usually, and you're going to have a full 8 histones, 8 histone proteins in here, and this octamer is what the DNA is actually going to wrap around. Because, oh no, I don't want that. Because this DNA is negatively charged, and these histones are supercharged with positive residues. Also notice here, look at all these tails flying off these histones. That is where you're going to be able to modify how well the DNA is binding. So let's take a look at that. So we'll take a look at that in the next thing. This is just another very good illustration of the subunits of each protein, how they're going to combine to form essentially this like really tightly wound disc that is going to attract DNA and keep it stable like that. These are just two nice ways to visualize how DNA is going to interact with these proteins. And as you can tell, they're kind of still in a, kind of that circular helix formation, but we'll kind of see how this can get a little more complex and if you want it to be more open, tighter, things like that. Okay, so the histone code. We kind of talked about this a little bit in class, how one of the big mysteries of biology, for example, is how to, predict the amino acid sequence of proteins and how that's going to fold. The other is that to unravel DNA, you can add little groups to different spots on the histones, and each one has different, effects, essentially. So there's all kinds of proteins that can add acetyl groups. They can add methyl groups. Sometimes there are phosphorylation additions that you can have at certain enzyme or at certain regions. So what these tails are showing you are lysines, and that's what the K is for, right? You have some arginines that can also be modified, but it's mainly lysines. So let's take a look at this one that I have right here circled. This is lysine H3K27. What that means is that it's the lysine 27 on the H3 histone. This mark in particular, this acetylation, this is very, very important for creating an environment that is very good for DNA to break free of this, of the positive charge of the histones. So remember that histones are super positive charge. This whole octamer is just, exuding positive charge. A lot of that is facilitated by these big lysines. Remember, these are charged. When they have an acetyl group like this, that is going to neutralize that charge, and it's going to release DNA. So H3K27 acetylation is a massive, massive mark for active chromatin, for example. Most of the time, like we talked about, when you add an acetyl group, you are breaking the hold that these histones have on DNA. You are opening it up. Most of the time, that is true. With methylation, we've kind of talked about how that's usually a silencing mechanism. That is half and half true. Methylations to a lot of these areas are going to silence DNA. Other methylation regions are actually going to serve as docking sites for big transcription factors, for example, that could come in and say, okay, we have our docking site because there is a methyl group here. We are going to actually start actively transcribing now. So, if you want the easy picture, it's that methylation can signal either way, and that acetylation is oftentimes the active signal. That is the signal that says, okay, this DNA is going to open up because we're neutralizing this K, this positive signal here. So the code itself is managing and deciding based on all of these, and this is only a small selection, based on all kinds of different alterations that you can make to the proteins that are going to affect the DNA's silence or active, basically, phenotype. There is a combination of these modifications that will lead to genes being opened up. Now, we are still playing with fire when it comes to these combinations. For example, I know one of the main cancer proteins that I've studied in my past is something that gets a mutation that usually adds acetyl groups to this group right here. And if you lose CREBBP, which is the name of the gene, you lose this acetylation, and all kinds of genes that were once on are now off. So mutating a single gene that's responsible for one of these marks can cause massive changes in gene expression, gene knockout, silencing across the genome. So if you mess with one of the master regulators, you completely change the cell's phenotype. That can often lead to cancer and follicular lymphoma in a lot of cases because you've basically knocked out or in a combination of genes that were relying on this mark. So again, if we could figure this out, we could rewire the genome with certain combinations of inhibitors. But like I've said, right now what we're doing is essentially sending in inhibitors that will target proteins that actually change these and then see the effects. So we're still very much messing with fire, like I've said a million times, sorry. So, kind of going around that 200 base pair number, it's basically just physics. At a certain point, you can do 1.6 wraparounds, and then you do it again, and you have this little open region. And so when you have more frequent open regions like this, then that means that this DNA is more accessible. But for the most part, yeah, I shouldn't get ahead of myself. Essentially, this is just the structure that you're going to see over and over again. The little nucleosomes are going to wrap DNA tight and keep it compact. Because again, like I've said, you have a lot of DNA, and you could stretch that out a long, long way. You need to compact it through a system like this. And so the 200 base pair thing, that's just because when we digest these individuals, like in this little range right here, it's about 200 base pairs. Okay, so we're going to go back to this slide. The tighter that this is packed, and as you can see here, so the tighter this is packed, the more inactive it's going to be. You just can't access it. So the looser this is packed, the more you can access. This is all open. A lot of these proteins can just go in and bind, if it's an open area, they can just bind DNA right here. And that's perfect, and then you start turning on genes. So these nucleosomes, this level of control is highly, highly managed. Okay, where things are active and where they're inactive is incredibly managed, or sorry, it is very controlled, I should say. Okay, so we talked a little bit about histone methylation and histone acetylation, how that works. So let's just revisit so that we don't get confused about this, because it's a good thing to revisit. DNA methylation is a different thing. It's in conjunction with the histones, but remember, you're not methylating. So if we're up here, these are methylations, these in yellow here, on protein tails, and that's going to affect transcription, silencing, inactive, active, because like I said, it can kind of go both ways. When you methylate cytosines, though, you're going to have something called DNMT, and that is going to take cytosines, and it's an enzyme. It's going to take a methyl group from something called an SAM, and it's basically just a carrier for methyl group, and it's going to add that methylase. There are other DNMTs that don't add the methylases, but they definitely maintain the methyl groups across the genome. So this is a way when your cell divides, this DNMT1 will come by and say, okay, the parental strand has a methyl here, I'm going to add it to the other replicated strand that we had over here and make sure that that methylation pattern stays the same. So DNMTs, these are going to add methyl groups to DNA, and that's usually going to silence that region. So remember CPG islands? Those are collections of Cs, so cytosines, and they're usually a gatekeeper for genes because if they all get methylated, that basically shuts down transcription usually. So CPG islands are a great spot to get methylated to turn the gene off. To demethylate something, you have TET proteins. Again, look in your book, and if you want to know like the big name and everything, it's actually a weird name. It's like translocation 11 like structure or something. It's strange. Remember, people name genes for what they're first discovered for, even if that's really not what they're really doing a lot of the time. All you need to know is that TET is the opposite. It is going to take away methyls. TETs are going to activate genes, for example, that have been methylated in their CPG islands so that the promoter can get accessed because it goes back to cytosines. A lot of the time, methyl groups honestly just usually go away at a certain time. They kind of have that half-life that we've talked about with so many other things. Methylation usually doesn't stick around forever, so you have to be maintaining methylation through something like DNMT1. But if you want to actively take a methylation off, you need to probably use a TET protein to remove that methyl group. Okay. So with DNA methylation, if you ruin the usual pattern, let's say, and you're not methylating the right stuff, you are going to unleash a lot of genomic instability. You're going to have genes that are on that should not be on. Maybe some of them are drivers of growth or proliferation, and that's going to give you a cancer advantage. So you didn't have to mutate anything here. You just had a very bad, you did not have enough you didn't have enough methylation. That's what hypomethylation means. And genes that got out and active were not supposed to get out and active, and they did. So here's a really good way to take a look at a CPG island. So usually this is a tumor suppressor gene. Remember, we've talked about these. These are genes that have any role in stopping excessive growth of cells, or they promote cell apoptosis, the cell suicide. These are very good to have if a cell is going bad. This CpG island here is marked as unmethylated, so that means it's active. If you accidentally methylate these, you will literally just silence this gene. That is as good, if not worse in a lot of cases, than mutating this gene, because now you have lost a transcriptional, or a tumor suppressor gene. So remember we talked about the angel gene, or BRCA1, for example, or 2. Those are TSGs. Those are genes that check the cell for integrity. They make sure everything's good, right? You don't want to lose those. If you have the wrong methylation in something, it's a very bad deal. So reversing that, though, and say adding some kind of inhibitor to whatever's methylating these, let's say you return this to a nice unmethylated state, this gene can basically rise from the dead. That's why we're messing with a lot of these things, is that we don't lose a lot of tumor suppressing genes to mutation in a lot of cases. Sometimes they're just silenced and we didn't know why. By changing the methylation state, we can reactivate them and, like I said, bring them back from silenced. So it's very good. So another fun point to kind of go over with, given certain circumstances and the viruses are very interesting, is that we have a virome. Humans carry around, and we've talked about this with the transposons, or the jumping genes that, kind of put themselves everywhere. That's a viral mechanism. It's an ancient viral mechanism that's kind of out of control and, kind of pops around our genome a little bit, but we've been infected with viruses that integrate in our genome for millions of years, and a lot of times viruses really don't do anything other than that. They just jump in the genome, and that's about it. A lot of the ways that we stop them from jumping too much is that our body has certain mechanisms that can find viral DNA and just be like, oh my god, methylate this, please. It's methylated, all of a sudden it's silenced, for example, just like in this example, and then we seal them away. So this is the principle, remember, of that paper that we talked about. Oh, yep, I talked about it right here. Nice. So these viruses in this virome that we have, that humans are composed of, in our DNA and our genomes are literally, a lot of that DNA is viruses that have been integrated. Methylation is the way that we control those species from just all arising. So when we added an epigenetic inhibitor, and we started to, hypomethylate a lot of regions so that we could get tumor suppressor genes back, for example, maybe, all the methylation for all these viruses was lost. A ton of them just awoken and started making viral products that, although they're totally harmless, they look very foreign to our immune system. So it was a very cool consequence of these inhibitors. This is just awesome. This was just a great slide that I came across as far as viruses. I'd hate to waste your time on it because it's not. I'm not going to talk about this on the test. I think as we get closer to some more nucleic acid, like is it alive type stuff, this is something we can revisit though, so it's not bad. But if you are, cramming for the test right now and you want to skip about 2 minutes, go ahead and hit that over key a few times. This is just super cool. Virus early, this is the one that I kind of subscribe to in a lot of ways because we existed in a pre-cellular stage already. So it kind of makes sense that nucleic acids that would copy themselves and then, kind of exist well like this, eventually found their way to infect cells and parasitize a lot of this machinery. That's interesting. This is a very cool hypothesis, basically that we had a bunch of protocells running around, and some of them basically left the cell phenotype, which is like, plasma membrane, complex structures and stuff, and they're just like, nah, we just want to put our genes in these other big cells, and we're good. And that's a very good survival mechanism. This one is awesome. This is the idea that there was a gene that could replicate itself over and over, and that it could basically keep replicating itself and then spread to other cells to keep replicating itself. And thus that was the first idea of a virus, spreading genes. Number 3, C is awesome. That's super cool. I think all these are super cool. So if you're interested in medicine, history of viruses and stuff, this is just an awesome set of slides. Really cool. And I love that we got to talk about this in person. So hopefully some of the, in-person meetings that we do with this class can come back to this type of stuff, which is, like I said, with the, the virome that we have, a lot of our genes mimic viruses, how they work. Like we talked about with this ARC protein that, when it's transporting stuff between cells, no, I can't draw too much. So it's like going to transport vesicles filled with receptors to all our cells. and it's behaving like a virus particle. And I would definitely recommend looking at the video that I have on Moodle for that, because it's just awesome. What's a human gene, what's a virus, and what does that make us? This has that combination of, trying to elicit that thought with the microbiome too, all the bacterial species that exist within us. We are very much a superorganism with multiple things kind of pulling the strings, to say the least. So it's a very interesting idea that organisms are kind of maybe a collection of organisms sometimes. It's very interesting. Okay. Oh no, back to histones. Don't worry, these are fun. Each site has DNA binding sites. Each site also has binding sites, so you have the DNA binding sites, right? You also have to have binding sites so that you can pair up with each other. So you can pair up with each other here, and so you can pair up with each other just as a whole octamer, right? And then the DNA binding sites are going to interact all along this string. What this is showing you right here is that the 4 core octamer type histones, they're pretty similar. And this structure, this DNA binding structure right here, is highly conserved. It's not exact. The changes in the red show that, there's a lot of divergence in some cases, but this is the key structure to storing DNA. This is something unique to eukaryotic cells as well. Bacterial prokaryotes have other DNA binding proteins. They're not histones though, and we'll see that they're much simpler, and that has certain advantages to being simpler, but you definitely can't form something like us with a simple mechanism like that, and we'll see why. So this slide kind of explains itself. The chemistry and the physics behind A and T double combinations, so usually it's a pair of A and T's right after the other. They most likely like to exist on the outside of the helix as it is, oh my goodness, I hate these, as it is binding to, there we go, as it is binding to the histone. Most likely though, in a lot of cases, you are going to have more GNCs on the outside. Yeah, oops, I'm sorry, I meant that it goes inward more. So as you can tell, there's a lot more blue contact with the inside of the histone, so like this is a really good set right there. Then there is outside yellow contact, which is the G's and C's. See how they're kind of mainly going to be put on the outside like this. Sorry, I misspoke quickly like that. I always do that. There we go. So yeah, take a look at that and just kind of remember that everything here has like a very big plan as far as fitting and how this DNA has to be structured. Okay, so as you can see, the DNA is going to wrap around these histones like that, and the histones do form a core in here, but it's these tails that are sticking out. These are the things that you can modify. These are very accessible to add acetyl groups to. You can methylate these, you can phosphorylate these, and the combination of those acetylations, methylations, and everything, that's going to have an effect on how tightly this DNA is going to stick around. Okay, so this is kind of a checkpoint. This is where you can pause, kind of remember that what I want you to know is the ingredients that it takes to open a gene up, the competing factors that decide if something's open or not. Okay. In each case, you're probably going to have an enzyme or kind of some master regulator protein that's going to facilitate these modifications. Okay. There are examples of true proteins that exist like these. So acetyltransferase, I talked about one that gets mutated and you lose a bunch of acetyls. That's called CREB-BP. There's one that's a partner called EP300. There's tons of these genes, and you will see a lot of them as medicine and everything else moves towards an epigenetic, focus. As we kind of get into a genome that is fully developing all the time. So as we go through, let's take a little focus here. And like I said, you can kind of pause this slide, and it's going to give you a lot of the info right here. When you have a histone acetyltransferase to show up, they're going to put on a ton of acetyl groups. That's going to be great because you neutralize this lysine, and that allows the DNA to unbind and become a lot more accessible. All these regions in here are hyper accessible now. Doesn't look like a lot. But if you remove these acetyl groups through an HDAC, histone deacetylase, all that DNA and the histones, they are now like super tightly wound. You are not going to get them. There's barely any room here compared to all this room right here. This is silenced because you're going to have a great all these lysines are going to be really positively charged, and they're going to bind that DNA right up. So the interesting thing is that hats and HDACs, they're often fighting each other in regions of genomic, not instability, but regions where active chromatin exists. So when you lose an acetyltransferase, let's say you lose one of these, the HDACs just take over because your cells exist in a certain balance. So if you lose one to mutation or something, the HDACs will just silence everything because they don't have an opposing force against them in a lot of cases. During one of our live sessions, I'll try and go over a really interesting phenomena of the hat that we study when it gets mutated in certain regions. Okay, so like I've said, DNA methylation is a pretty surefire sign that that DNA is a silenced region. That's how you silence viruses, that's how you silence CpG islands in the promoter, things like that. When it comes to methylation and phosphorylations, though, of histones, there's some leeway to what's going to happen in a lot of cases. There are a lot of methylations of histones that are going to silence them, but there's a lot of methylations that you're actually going to attract transcription factors. As you can see right here, this transcription factor is attracted to the combination. And remember, this is why it's a histone code. You need a certain combination to attract these TFs by the combination of a phosphoryl site at this histone and a methylation site here. And it takes a big combination to allow that. So just right here, for example, if you only have methylation groups like we have, and we've removed one of them up here, that TF can't get in. See, it's kind of like floating up here. Here, it's all ready. That's bound the DNA. If it can't access the DNA anymore, you're out of luck. And this gene is not going to receive the transcription factor. It's never going to turn on. Okay, so for the most part, you are going to... we should go here. So I should highlight these. So we highlighted HAT and HDACs already, right? Those are going to do acetylations, and those are pretty clear. Protein kinases usually add phosphorylation groups. Protein phosphates are going to, they are going to remove them. Now, kinases and phosphates, these have a ton to do with like the actual cell signaling that you would see in something more like physiology, the cascades of signaling, all that. In histones, they kind of have a variable role. So HMTs are going to add methyls in this case. And HDMs, or histone deacetyl demethylases, those are going to remove methyls. So they kind of work against each other. Remember that each thing kind of has a partner that takes away whatever it added. So with methylation, in a lot of cases, it's going to repress stuff. Because this methylation, for example, is going to attract repressive proteins. That's the key, is that this altered amino acid right here is going to attract specific combinations of proteins that really just shut everything down. And if these proteins show up because of this signal, everything is just done, basically, because these keep the histones really sealed up. Now, it can go both ways, though, and again, this is why it's histone code, is certain regions, when they're methylated, are actually going to attract big complexes of activating proteins. So you can see here, under the right circumstances, so see right here, this is H3K4. When it's methylated, it's going to activate because it's going to bring TAF here. But if it's H3, so histone 3, lysine 9, if that's the one that's methylated, you get repressed. Same thing, good example over here. So you can tell that the interplay between activation and repression is going to depend on what histone you're modifying in a lot of cases. That's why this code is so complex, is that you're having to deal with basically what modification's happening, which one of these histones is it happening to, what's the effect, what's it attracting. I'm not going to need you to know the specific regions and everything. Just know that there is that fluidity. Okay, so as far as, remember, now we're back at DNA, so we're off the protein modifications. This is just kind of a checkpoint to what we talked about. DNMTs are going to add methyl groups to this cytosine. See, unmethylated here, DNMT, and you're good. You showed up with a methyl group. And now this, as you can see with the dotted line, this gene is now pretty much taken out. So TETs, what they do is they initiate a process that is going to lead to the loss of the methylation group. So the first thing they do is actually they add an OH here. That OH is going to transition through the facilitation of another TET or the same TET to a double bonded oxygen. And eventually you're going to go right here. And then there's a final step for something called TDG. It's kind of a partner of TETs, but it's going to basically just finish the job so that you can basically, basically go back to an unmethylated region. So TETs are going to activate a lot of genes, DNMTs are going to silence a lot of genes. I included this slide just because it's kind of, it's helpful. You can pause right here and take a look. This is from a cancer paper, Cancer the Journal, so it's a good journal. And it kind of just goes through how everything works. In a lot of cases, if you have this methylation, remember this was one of the active methylating groups, then you are going to, in a lot of cases, or no, that's actually one of the, that is the, oh yeah, that is one of the activatings. See, it's hard. I'm not going to ask you to memorize that. That TETs and DNMTs are going to fight each other off, basically, in a lot of cases. And that TETs, a lot of times, actually occupy the region that's unmethylated for a gene so that transcription factors can come and find it. So that's actually another role that has been found out about these. Not only do they keep the methyl groups off, they literally, the protein keeps them off as well. Okay, like I said, this is in blue. We're talking about individual, modifications at these little tails. The other side of that is that these modifications aren't just going to control, interactions with this DNA wrap. They're going to be interacting all over the place. They're going to be crossing paths, and at different modifications are going to interact with, different combinations of the tail, basically. It's complex because you're dealing with something that is super intricate, and these chemical reactions are, binding to each other all over the place, basically. So, hey, that's why it's a 300 level class, right? Okay, kind of this forgotten histone, H1. H1 is not part of the octamer. It is called the linker histone because it mainly interacts with the DNA that's outside the histone octamer. Now, the important role that H1 has, and we'll see how this can change, how you can move its position, is if its position is occupying a lot of the DNA, I think kind of like in this one, there's not a lot of free DNA around here, versus over here there's a whole stretch of free DNA. It really depends on the position where H1 is in. So like we saw in that graphic a long time ago, if you have an H1 that's binding this DNA, a TF can't come in and actually bind there. You need to remove H1 before the TF, oh no, it's an ugly T, the TF can actually bind. It's a really ugly F too. So H1s can, a lot of the times, even though you may have freed up some DNA through acetylation, if an H1's in the way, it's probably going to block all these TFs. Oops, can't delete that. Okay. So again, now we're in another genomic mystery of science. What these are is the models of how nucleosomes exist in compact states. Each one of these little models right here is a nucleosome with full wraps of DNA. So each of these is a dense packing of DNA wrapped around these histones. So if I draw it like that, it's more like something like this. And these are essentially supercoiling around each other to compact. And you can see that here, how narrow that is. And you can see the little histones right here, how they're stacked just right on top of each other. This is one model. The other is even more, beautiful to say the least, a helix of nucleosomes, an interaction between nucleosomes that would make the complexity of the code so much more difficult to understand someday, because you would have to be looking at what controls the strength of this helix. How many times do you have a large bubble come out, and is that region, more open active transcription DNA, things like that. Personally, looking at this and reading on this, it's very, to me, it's almost that maybe states like this exist in silenced regions, and both sections are wrapped up like this, and maybe more active regions could have the solenoid model. So that's something to think about, if that makes sense with everything we've seen. You can join a lot of the other thousands of scientists that still don't have a true answer to this hypothesis yet. But we know that it's some kind of structure like this. We just can't confirm it between the two. But it is really, really cool, to be honest. Okay. So the other thing is that if we are looking at a solenoidal model or the helix model, the zigzag, the pitch and the, kind of the amount of DNA that's wrapping around is going to change. The space between can also change, as you can see. Like can you see how these are changing? This one is facing us, and this one we're looking right at lateral, right? That is a whole nother, thing that you can cite about the dynamic nature of nucleosomes, which is they can be facing different directions too in their 3D shape. And what that means is that different tails coming off these histones may only interact in certain conformations. Let's say it's going to go to this one over here, or it's going to go to this one. Oh, I'm ruining it. Let's say that this tail is going to interact with certain others in a 3D shape that's only going to be facilitated by certain shapes here. This is hard stuff, yeah. So, this is good that this is almost good that this is a video lecture that you can pause and kind of take a look at. Really do try and read your books, people, by the way. Like, I understand that books are not popular. You're not going to find the answers that some of you need to build associations without that book. We're going to have some live classroom time for this, and that's where we're going to be able to kind of make some connections and go through things. But that book's a great resource. I understand nobody likes buying books, but I know. So this is just a really good look at a helix of nucleosomes and just how tightly packed your DNA can get. 4.7 billion nucleotides in every single cell, and if you stretch those out, I think it's something like the distance to the moon or something. Maybe I'm being hyperbolic, but you should check. It's huge. And that's why you have to have this gigantic, spooled, amazing structure to keep all this DNA silent, basically. Okay. This is just a very good slide showing kind of what we have gone over and how compact everything gets. This is also going to tie very well into Topology Chapter 9, because we're going to see these rosettes again. When you get these massive filament fibers, they are going to supercoil in these massive either solenoidal or helix shapes, and they are going to be on a scaffold. See this right here? That scaffold is keeping all these histones and nucleosomes set up like this. That scaffold is just part of the rosette structure. Remember these little green and purple things? Remember how they hook together and they form this amazing rosette structure? These rosettes stack upon one another, and that is when you start seeing. That's what you're seeing right here are the rosette structures. The rosettes are formed of these protein linkers, and they are facilitating massive helices of nucleosomes that contain your entire genome. Well, this is one chromosome, but still. I think it's pretty cool. Okay, this is one of our first differences with bacteria. This is literally what the bacterial genome was going to do. It's going to like kind of pile in these small little pockets, because remember, this is a big circular genome. It is going to supercoil like this, but this is just pure DNA right here. This is wide open. These are the nucleosomes. These are the histone-like proteins in bacteria, and they're not really making it that much more complex. Remember, these don't have as big of a genome as us. Remember that they also need to have access to their DNA immediately, should they need it. The minute you put bacteria on a surface that it doesn't like, it will start turning on this gene, it'll start turning on a gene here, turn on this gene, and turn on this gene. As good as it is to have your DNA stored, if you're a complex organism like us that has tons of genes, but different types of cells and you don't want everything on, a bacteria is very simple. It needs to react quickly. It needs all of its genes active. They need to be ready for transcription. They need to make a product immediately. And that is one of the biggest reasons why they don't compact their DNA into the same histones, is because this needs to happen immediately. You need to turn on this gene right now. Okay. This is fun. This full paper is, I think, Diaz 2013 in our Moodle. No, Diaz was a different one. Anyway, this is epigenetic inheritance. This is kind of what I was babbling about once, where if a parent has PTSD or depression, they are likely, if not predetermined almost, to pass on the epigenetic wiring that dictates their genome to their offspring. This isn't true for every complex disease or condition, but you can test this idea. In this case, you feed a mouse cocaine over and over and over again. That's the increasing dose of cocaine that this parent mouse wants. That's a lot of cocaine, I gotta be honest. It's a pretty stark experiment to how much this mouse gets used to cocaine. it develops a straight up resistance in its brain. Because remember, cocaine's a very quick, stimuli activator. The brain rewires so that when you have cocaine, it doesn't break, essentially. It makes it so that it's a normal state. Now, the authors don't go as far to say that this mouse is addicted to cocaine, but you definitely have some modifications that have led its brain to not basically react the same way. Now, the cool thing is that it will have progeny. We will measure these progeny here. They will have never encountered cocaine, but the minute that they do in the lab, they have the resistance, and we can tell. We can look at the methylated or active acetylated regions, and we see that they have been passed from the father. And note, again, it's a father, not a comprehensive experiment, to his offspring. Okay? You can pass on addiction, stress, all kinds of different things. You really do get more from your parents than you think in a lot of cases. That's not to say that you can't break epigenetics. You can. You really can. I do believe that. In some cases, you don't, though. And so it's kind of there's kind of some gray area as to what you can have. But this is one of my favorite points from the class, which is that, and I kind of asked you, nature versus nurture. It's both. It's always going to be both. It's a developing genome. This genome responded to the more cocaine that this mouse took in, and that genome was passed on in its wired active or inactive state of those genes to its offspring. It's pretty cool. Okay, another really good way that you can decide what regions are open is you can remodel where the histones are at, for example. So in this case, let's say you start right here with this histone setup. There are a lot of ways that you can change this. There are enzymes that can push this histone over to here, and now you expose a big DNA region right here. You can straight up delete histones sometimes, and now you have exposed a really nice genome right here, or like a nice piece of DNA right here. So you can remodel what your epigenetic landscape looks like, and enzymes are going to do this in a lot of cases. You can go from this where genes are inactive to like, okay, we moved it, now this gene's active. There are also variations and isoforms, as we call them, of the histone proteins. Okay, remember how mRNAs can be differently spliced. Sometimes the same thing happens with histones, where you can insert a different piece of the octamer, and sometimes this is going to bind DNA better or worse, and that's another way you can change it. So modifications itself, you can basically make a cis modification here, and it's going to open everything up, or a trans modification in a lot of cases. What trans modifications can sometimes do, and remember, read your book because these can kind of bounce between what they actually do. These are going to attract proteins that bind to specific signals, and these proteins can have different effects. Some proteins that you attract are going to be very, they're going to shut things down, but a lot of other proteins that you attract can actually cause, gene activation. So it's really going to depend on what protein, what signal is going to attract. That's why this modification slide is so simple. It's basically saying different modifications are going to attract different proteins that are going to elicit different effects. This is kind of a cool little technique. It's in green, but essentially this bead is going to stretch a chromatin fiber, the ones that we've been talking about, onto another bead, and we can actually measure the stretch here. So this force that's elicited here can go a lot higher if you have open DNA versus if you have super closed DNA. That force before it breaks is really small. So here, based on this force, you can be like, okay, this region that we're taking a look at, this is a closed region. Versus this region we're looking at, okay, this has an open gene in it. This is something we could study. Okay, so this is the application of remodeling those chromatin, those nucleosomes, okay? If you get one of these big complexes, and remember, this is a big collection of proteins usually. This is not even a single player a lot of the time. You're going to have like 4 or 5 bubbles of proteins that come in and they remodel stuff. We'll see it in the next slide. But if they come, they can unwind the DNA. So again, these are managing topology here to make sure this DNA is wound nicely. And they're going to basically unwind some DNA and get this whole space. So see where we started here. This promoter's tucked away in the histone. It is completely inaccessible. It's not even in the linker. By moving the histone, though, you've exposed the promoter, and now you can activate that gene. So how these work is they basically grab a histone, roll it around, and you can reposition the DNA. That means they can position the protein or they can reposition the DNA itself. Doesn't really matter. This is one of my favorite players in DLBCL, the cancer that I study. The SWF-NF complex remodels histones. That's a kind of a goofy name because these genes, again, were discovered, I think, in fruit fly, so it's another goofy name. You can look it up if you want. It's like something about sucrose or I can't even remember. What it does, though, is that if you have a gene that basically is stuck right on the octamer, see how there's 8 right here? If that gene region that you need to access is stuck right on the octamer, it needs the help of a complex like this to come in, and it basically unwinds a little bit of DNA from the histone, and now the transcription process can start. Also, I know I'm like pretty loosely talking about transcription and promoters and stuff. If you guys have any questions on that, you really need to let me know, because that's a huge part of the test, is basically that I assume you know all that. So use one of these meeting times coming up to do that, please. And literally, any meeting time is literally any meeting time at this point. Okay. So we have really good ways to figure out where active promoters are in a lot of cases. Active promoters need to be outside of a histone. They can't be bound up by the histone protein because you need to be able to get TFs that are going to attract RNA polymerase and that's, make RNA for you because that's an active gene, right? So they need to be exposed. This is another nice example right here of a nice exposed region. What we can do is measure the occupancy of nucleosomes along the genome. So look at all these base pairs. Here is a little spike right here. See that? That spike is a dip. There is not very much nucleosome occupancy right there, right? Now, when you have really tight sections, these are in nucleosomes. These are positioned on nucleosomes. That means they're super tight. There's a ton of occupancy. Remember this dip right here? That translates to a really tiny occupancy of nucleosomes at that spot. So when you go to the USC Genome Browser, you can open up tools that show nucleosome occupancy, and you'll be able to see regions that are open. They are open for business. They are open to transcribe that gene, basically. Okay, so the way that we can do this in a lot of cases, remember ChIP-seq? That's going to bind up a specific protein and see what DNA it's binding to. Perfect, but do it to the histones. All you got to do in this case is cross-link everything, so you fix DNA to any protein it's on. You digest any DNA that's not fixed to a protein, so in this case our histones are still carrying proteins. Now we get antibodies to our histones, and so these antibodies, remember, they're specific to histone 3, for example. One of the most specific antibodies I used to use was histone 3 lysine 27, because then I'm only getting histones that are bound with that mark too. So this can get even more specific. If you don't know what antibodies are or what they do, again, reach out to me or watch some videos. You really do need to know that. Okay. So then at this point, if you've isolated the protein that the DNAs are cross-linked to, then you digest the proteins, and you only have DNA that was bound to nucleosomes. Now, again, this is Sanger sequencing. That's boring, and that's going to take a long time. Just put that thing on an NGS panel and sequence the living hell out of it, basically. Sequence all the genes that were bound by histones. Those genes are the ones that are closed for business, okay? Anything that is bound up tight by a histone is not active right now. And this can change cell by cell, remember, in a human. You could take a look at this pattern right here and compare it to, intestinal cells versus a heart cell. You'd see a huge difference in what genes are active and inactive. So that's great. Okay. Like I said, more complexity. But remember, I have open questions. So if I ask you why is the histone code complex, you can toss in all these reasons that we've talked about in this video. That's literally probably a question I'm going to ask for those of you that have made it far enough in this video. When you get, into this complexity, it's because, look at this. There's a variant of H3, for example. So there are protein variants that have different modifications. Different histones have different modifications that are going to attract different changes, and that is super complex. And like we saw with H2A, there's not a lot of defined things. H3 is the one that we've studied the most, and it has the most definable alterations that elicit effects, these methylations, phosphorylations, and some of these acetyl groups. Okay, so this is a very good slide because this is showing that sometimes when you have an exposed promoter, for the most part, if you can have an open space right here, that means you can turn that gene on. So that's good. And in a lot of cases, there are certain proteins that actually lock these in. They lock in this state so that no remodeling complex eliminates the promoter. That's what something like H3 is, for example. This different type of histone has a tail right here. See that little tail? And see when you make octamers out of them, they lock the nucleosomes in this position. See how this tail is a little longer? No, it's not going to show it there. See that big blue tail right there? Instead of the typical histone tail, H3.3 is something that these protein tails now are going to interact with each other, and they're going to basically lock in that state. So that's really cool. So that's a way that you can rewire things, is tell your genome, pull all the H3 normals out, put H3.3 in, and it's going to basically keep this state active. And as you can see, the old promoter, just regular H3, just gets kicked out then. Okay, so another thing you can do to modify these nucleosomes, you can put in something called SEMPA and exchange it for H3. What SEMPA is essentially, it's an H3 variant, but it has a massive protein tail. These are great at attaching to kinetochores. Remember, kinetochores form when you're dividing the cell. They're what the cell pulls on to pull the DNA. So when you're pulling this massive amount, you need something that can hook on. And that's what this variant is right here. This is another very important one. This is very cool. When you have a double-stranded break, one of the mechanisms that detects this break is going to send in this H2A variant. See how the H2A goes away? It is replaced by enzymes by H2AX. H2AX is amazing and has a lot of phosphorylation sites that it can get phosphorylated by. These phosphorylation sites are going to immediately attract DNA binding and repair proteins. So when we talk about something that's like, oh, this detects DNA double-stranded breaks, this is chemically how it does it. It finds the breaks, it puts in a new nucleosome subunit, that subunit gets phosphorylated, those phosphoryl groups attract repair proteins, and they seal the break. Pretty cool, huh? This is a long lecture, huh? Okay, let's take a look here. So we talked about how we can just exchange these little pieces, right? Like we've been exchanging these proteins in here. There are certain chaperone proteins that make that happen. That doesn't just happen on its own. Certain ones are going to facilitate the addition of H2As. Others are going to facilitate the addition of H3s and H4s. And like I should have said, H2Bs. So specific octamers and formations are facilitated by specific remodeling proteins. Okay. This is just a good supplement slide showing all the regions that we know to have some kind of effect. As you can tell, H3 is the busiest. Where's my 27? There it is. There's HK27. Oh, I ruined it. H3K27. That's a big deal right there. Shows really open chromatin. Sorry, I have all these details. Okay. Again, kind of a nice supplement slide for the chemists in the room. These are the effects that you're having when you add acetyl groups, or you add methyl groups to lysines, or when you add methylation to arginines. And there are variations of it. For example, let's get into this, for example. So methylation of lysines, a trimethylation may attract certain proteins to come in and bind that region of the nucleosome, or maybe it's a signal for remodeling. A single methylation could have a very different effect, for example. Okay. This is where things get a little different too. So let's say that you have really open active chromatin. You have a ton, so what these are acetyl groups right here. And this is a nice exposed promoter. One of the ways that you make sure, so remember we saw the nucleosomes that can kind of lock into each other. You don't always need to do that. You could have something called a bromodomain protein. And remember, you don't need to know the specific names, but I mean, it doesn't hurt for your understanding. It'd probably make it easier if you did, but all you got to say is that there are proteins that basically bind acetyl groups. See how that nice purple is in there? And it keeps them there. It protects them. It does not allow things to exchange them to inactive. See, because up here, if you don't have protector proteins, an HDAC could come along, a histone deacetylase, sorry, there it is, and it could just return it to the inactive state. And if you don't want that, bromodomain proteins are a great way to ensure, okay, we're going to stay in this active conformation as long as we need. So as long as bromodomain proteins are around, this is going to stay awesome. Okay? Chromodomain proteins are the opposite. They are going to find methylation groups and they're going to say, we're going to bind right to these and nothing's going to move. we are not going to allow it to open. Okay? Stabilized closed state versus a stabilized open state. Cool. So with these, we are still figuring out what motifs in the histone code attract certain proteins, and oftentimes what their categorical function is. So this table does a good job showing there are different answers for each modification in a lot of cases. Some are transcription, some are silencing, for example. So it's very interesting to see that. Remember, SEMPA is the one that is going to be good for kinetochores, so it's associated with mitosis and phosphorylation. Yay, almost done. I mean, there's tons. So basically, all this slide should have said, this should be in blue because it's a good summary. There's a ton of ways that you can hide promoters, that you can keep promoters exposed, that you can re-silence them or something. And these complexes are incredibly fluid at moving the nucleosomes around so that you can expose DNA, for example. Then you can get the main transcription binding stuff. So this is a TF2D. This is a main transcription factor. And then you can expose it because you have that open promoter right there. So moving these little nucleosome octamers around is a massive way to change things, and it's really cool. And we are still learning so much about this. Like, we know the basic targets of a complex like this, but mapping this in every different cell, that's going to take years. But it's going to be awesome when we have those answers someday. So we talked a little bit about epigenetic memory. What this is when you replicate DNA, half of your histones are going to make it, and they're going to be like parental histones, basically, and they're going to get marked as parental ones. So basically, then you can have a bunch of marked as parental H2A and H2Bs too. Those get incorporated alongside the new H3H4s and the new H2A H2Bs. So the original parentals that you're really going to have are going to be the H3H4s and their state, and if they're acetylated, methylated, whatever, that'll get copied. So essentially, they're going to form new nucleosomes along the building section here. And so remember, these are strands that are coming along. These are the same strands. So you're going to add one here. You're going to add one here. As you can tell, this is a completely new one. This one is a new H3 combo with H4, but it's also one of the old H2Bs. So the H2As and H2Bs, they shed, but they can get recycled, versus the marked, kind of the ancient H3, H4s, they're the ones that signify, okay, here's the position we used to be at, adjust accordingly. They're the ones that are the markers of the parental strand. So that means as you're making like a new strand, for example, these are the ones you're going to follow. So your book does a good job, again, going over the fact that you can have 4 different combinations then here. Totally new, totally old, and you can see the shading there. Half and half, so a new H3, but an old H2s. And then I want to find one. There's one. New H2s, but an old H3 set, for example, H3 or H4s. Complex stuff, yeah. So pause it and ask me questions, because if nobody emails after this, I'm going to be a little confused how you have 100% understanding from one viewing. Okay. This can get maintained. So like we said, we just passed along these histones. So you're going to replicate, and now you have formed your nice new nucleosomes, but you have to pass along the acetylation that was there. These bromodomains are going to find acetyl groups, and there are proteins that are going to add one right next to the nucleosome next to it. Same thing here. It's going to recognize the acetyl. It's going to add one right next to it. And that's going to maintain the previous acetylation state that you had up here, right? Same thing when you have repressive methylations, let's say in this case. You're going to have these methylations here. You're going to tag them with those chromodomains. Those chromodomains are going to activate methyl transferases to come down, and they're going to add a new group of methyl here, and you maintain the epigenetic memory that all your cells had. So, very cool. We're going to apply a lot of this to some of the primary lit. Reread that virus paper, or at least the main concepts. That is chock full of this information, but it applies to something very disease specific, or no, sorry, very broadly disease, because it's talking about viruses, it's talking about cancer, it's talking about epigenetic disease in general. All of the stuff that we've just studied. This goes into that interaction of the developing genome. When the environment hits you with something, your genes like these, they are reacting. They are changing what is on and off inside of you. Okay, they are uncoiling nucleosomes, for example. They're exposing promoters by getting rid of H1. They're modifying histone tails to interact in a different way. They're methylating DNA. Okay, this chapter is so important because this is how you understand, you know, your genome is a fluid thing. It is changing in response to your current and chronic environment all the time. Okay, that's how people change in a lot of cases. And I know that people are, humans are very special, but when you change, don't be so sure that it wasn't your DNA coming on or off changing or that dosage was changing. There's a lot of things that can change just how much genes make you who you are in a lot of cases. All right, so like I said, the midterm is probably going to stop here at chapter 10 because I really want to focus on this, and we'll go from there. I expect lots of questions. Please email me questions. We should meet. I would love to see everybody and make sure that you're doing well, being virus-free. Okay?